Good morning, Year 9. Here we are looking at the second revision lesson out of 3 for the topic bioenergetics, and we're still looking at photosynthesis. So last revision lesson we looked at photosynthesis, and we're still looking at that today, and then in our third lesson we're going to look at respiration. So as I said before, my class, you've had these sheets. They are attached to the flashcards, the B4 flashcards that I put show on my homework. If not, then you can just recreate this in your book or on paper or however you want to. If you want to use some flashcards, make some flashcards, cut some paper down, then that's fine also. Okay, so this is the sheet that we're completing today. We're going to annotate all over this using ideas that are discussed in this video. Okay, so the first thing we talked about is we tend to say that plants undergo photosynthesis and humans undergo respiration. That's not really true, okay? All living things respire, and the rate at which that respiration takes place in a plant doesn't really change, and we can see that by this line here, okay? So respiration day or night remains pretty constant. However, photosynthesis, when there is a very high intensity of light, will increase. Okay, so at night the photosynthesis drops out, and we can see here this is probably dusk and dawn, and we see that in the height of the day, when there is the most sunshine, we've got the greatest rate of photosynthesis. So I think that's really why we kind of get stuck in this trap of saying that plants do photosynthesis and humans do respiration, because most of the time we can see that the photosynthesis actually far outweighs the respiration. Now this graph links to this graph over here, and we looked at this in class, and we were saying, okay, well, if we are looking in a greenhouse environment where the gases can be contained in that area, what will we see at different points in time? Well, at night time, when photosynthesis completely drops out and stops taking place, we will notice the respiration more, because the respiration is producing the carbon dioxide. So we can see blue line, carbon dioxide at night increases, and it's using up oxygen. So we can see at night that the oxygen decreases, okay? That's if we've got our gases contained in that greenhouse, we will notice these changes. Now in the daytime, where we've got photosynthesis so much higher than respiration, we notice the impact of photosynthesis much more than the impact of respiration. And as we know, photosynthesis producing oxygen, we see that the oxygen in the daytime increases, it uses up carbon dioxide, so we see that decreasing in the daytime, okay? So it's our way of kind of comparing the rate of photosynthesis compared to the rate of respiration at those different points in time, okay? And that's really important to remember. Respiration constant in the plant at all times. Photosynthesis is really only taking place when we've got that light energy there for that to occur. Now the other experiment that we looked at, which was not a required practical but really important, and one of the kind of exam board favourites, is the use of a potometer. Okay, we're looking at the bottom of the screen now. Okay, and we had 2 types of potometer. I've put in the most common one here. This is where we've got this air bubble here. Okay, now water is going to escape through the leaves by evaporation. Okay, remember that the water moves through that plant, through the xylem, by transpiration. So when the water evaporates, we can call that evapotranspiration. And just like when you drink through a straw, as the water escapes, more water is drawn up. So if we had this set up, we could maybe put a cold hairdryer on those leaves, and we could see in a fixed period of time, let's say 10 minutes, how much that air bubble moves to the left. We could put a ruler here to measure it, and then we could reset the air bubble using this reservoir here, move it back to the start position, and maybe we could change the the variable use a warm hairdryer and see how that affects it. We could compare different species of plant. We could compare different numbers of leaves on the plant, okay? But what we're using it for is to measure the amount of water that escapes through those stomata in a fixed period of time. I'm going to say 10 minutes, to see how that varies with different independent variables: wind, temperature, number of leaves, species of plant. The other kind of pedometer is a mass pedometer or a weight pedometer, and these are used where it's kind of the same thing. We've got maybe a pot plant and we put it onto some scales. We put a carrier bag around the pot so that no water can evaporate out of the pot, and as water evaporates over time, we see the scale reading decreasing. Okay, so it's kind of maybe a longer investigation when we might look at it over a few days, and we can change the conditions, maybe put it in different temperature environments and see where the most water is lost. Okay, so two types of pedometer there, not a required practical but quite a favourite. Okay, and it's investigating transpiration rates. Okay, that's the movement of water through the xylem. Now, it might be that at this point in time you want to have a pause and just leave that page up while you're working on your poster, on your revision sheet. If you need to pause, pause now. Okay, we're now going to look at the practical we did. Again, not a required practical, but a favourite with the exam board. This one is looking at the storage of starch in the plant. We said that plants have... they go through photosynthesis, it makes the glucose, and then they use the glucose in 5 different ways. And we did this in the last revision sheet where we said scarf, and it's starch, cellulose, amino acids, respiration, fats and oils. Now, when we delve into looking at the starch one in more detail, we can look at this practical, okay? So plants do their photosynthesis, they make their glucose, and then during the daytime when it's light, they take some of that glucose and they store it as starch for when it is dark, or even when it's winter, and then they use that, they break that starch back down again to use the glucose when there is no light. Okay, so glucose is the small unit, the monomer, and we join lots of those glucose together in a straight line and we get starch. And starch can be tested using iodine solution, and iodine solution starts off orange. If it goes blue-black, like we can see here, then there is a presence of starch. And we've got 2 things to look at here, really: where we found the starch and where we would find it in a variegated leaf. So we had, if you remember, a plant in the dark, a plant in the light, and the one that had been in the dark, we hypothesised that it had broken down its starch store, used it as glucose, kind of like a midnight feast, we called it, okay? And so here's the method. We boiled each of the 2 leaves. We then, and that broke down the cell wall. We then put it in ethanol, and the ethanol drew out the chlorophyll, and that enabled us to see better the results of the iodine solution test. And then we put it onto the white tile, we covered it in iodine, and we could compare the plant that had been in the light, the plant that had been in the dark. The one that had been in the light, we found did contain starch, which makes sense. It was still photosynthesizing all that time, producing the glucose and converting it to starch. And the one that had been found in the dark, we found that it had broken down its starch store, and the iodine solution did not change colour. Now, on this picture here on the left, they've done something quite similar. They've taken the leaf, and rather than putting the whole plant in the dark or in the light, they've just covered up part of the leaf, okay? And it is actually enough, and it does work, whereby that part of the leaf breaks down its starch, whereas starch still remains in the parts of the leaf where photosynthesis has been able to take place. And the final piece of information here is a variegated leaf. We saw that a variegated leaf has these white or yellow patches. There's no chlorophyll present in those places. So if there's no chlorophyll, photosynthesis can't occur there. So we never see any starch in those areas. So you can see here when we've done the iodine solution test, we've got no starch in those white or yellow regions because there's no chlorophyll, therefore no photosynthesis can occur there. Okay, so that's talked through everything for page 2. You've got the rest of the lesson now to fill in that sheet and revise that sheet. You've also got the flashcards that were on Show My Homework, and next time we're going to look at respiration and we're going to look at this sheet here. Okay, so I hope that went well. I hope you understood all the ideas. If not, have a look through your book, see if you can remind yourself. We did quite a detailed explanation on this one in our books. Okay, have a good day. Thank you.